The cell's response to the signal is to produce a receptor to accept more signals, to create more signals to be shared with nearby cells, and to access and modify a certain gene. How does the environment make an influence? The density of the cells in an area can affect the number of autoinducers produced. Hox genes and their roles in development. Homeobox, Hox genes, determine the physical arrangement of the body parts of an animal. Hox genes are grouped in clusters based on their shared chromosomes, and each cluster codes for a different part of the organism. Hox clusters are first activated when an organism is still in early embryos. They are activated in sequential order until the process of building the proteins are complete. WNT proteins act as ligands to start each portion of the process, for they carry the signal and information for when each cluster should start the process of construction. Without the WNT protein, the genes would not be activated. Although Hox genes are not activated until they receive a signal from the WNT protein, they are always in sequential order. The type of communication used to activate Hox genes is endocrine. This is because the information has to travel through the bloodstream and into the embryos. The gene can also be accessed later in the organism's life, adult years, when the brain sends a signal for its release. Hox genes do not have the information for the characteristics of the organism, for example, their eye color or their hair color, but for the pattern in which the physical trait should be. A great example is used in flies, for if the Hox gene of its antenna should be replaced for a Hox gene of its leg, then the fly would have a leg growing out of its head. The smallest change in the sequence of the Hox gene can have serious consequences. The physical placement of the fly is dependent on where each gene is expressed. Cytokines regulating cell replication and division. Cytokines are small molecules secreted by larger immune cells, T cells and B cells. They are used as signaling molecules so different cells can communicate with each other and are most often used in immunity. For example, a cell that has ingested a pathogen will send out cytokines, which carry information about the pathogen, to activate a helper T cell to bind with the antigen-presenting cell. The helper T cell will take this information from the antigen-presenting cell to a B cell, where they will bind via receptors, and it will release cytokines. These cytokines activate the B cells and give them information about the pathogen, so they can either spit into memory cells or antibody-secreting cells. This is a basic part of immunity. In cellular replication and division, cytokines are signaling/activation molecules secreted from an immune cell that tells a molecule to perform a certain task. The signal is normally secreted to pass along information regarding the pathogen so a cell can identify and destroy it, produce antigen fragment receptors, become memory cells, so if the pathogen enters the body again, the cell can identify it before it infects a great amount of cells, or that produce immunity receptors. This process uses the juxtacrine technique to communicate. Cytokines are produced as signals for other cells to start carrying out certain functions. Sources: